Good morning. We're going to finish up chapter 7 with this third video. The primary topic is going to be the evolution of life and the first forms of anaerobic metabolism in bacteria or organisms that basically, lack a membrane bound nucleus and other such organelles. Let me get the cursor right so we move from one slide to the next. So, science has an explanation for most anything. Whether correct or not, hypotheses are put forth, they're tested, theories develop. The theory that's been developed for life on Earth is called abiogenesis. And the mechanism of abiogenesis is called spontaneous generation. It's considered to be possible for spontaneous generation to have occurred in an environment lacking oxygen. And it's considered, pretty much 100% supported that when the Earth first formed, its atmosphere was void of oxygen. It was anaerobic. So oxygen developed later, and aerobic organisms developed from that. So you should assume that the first metabolic processes should be anaerobic, and that is the case. It's considered that at least nearly 4 billion years ago, there were prokaryotes, bacteria, and archaean organisms that were living here on Earth. This process of the formation of life called abiogenesis, one hypothesis for this formation of life that's been proposed is called the primordial soup hypothesis. It was developed independently by two scientists, Oparin and Haldane. Alexander Oparin was working and put forth some ideas from his home in Germany, and John Haldane was working in England. Oparin put forth his ideas first, Haldane second. They were very similar, so they've been combined today. The primordial soup hypothesis of both these gentlemen's concepts was something like an energy source, maybe lightning, sunlight energy, but mainly lightning, might have caused the energy needed to form small organic compounds. We call them monomers, things like sugar units or amino acids, maybe nucleotides. Oparin and Haldane proposed that these monomers may have accumulated in a shoreline location, or maybe deep underneath the ocean in a crack or a vent where ash or smoke was being released, or maybe near a volcanic vent close to the ocean surface, like you might find off the coast of Hawaii today. So these monomers accumulated, and with continual inputs of energy, they bonded together to form polymers, things such as a nucleic acid, maybe RNA is thought to be the first nucleic acid, or maybe even a protein, maybe an enzyme, or even a complex sugar, maybe something like starch or glycogen. From the organization of these polymers, it's thought that eventually a cell with living properties, metabolism, reproduction, inheritance, would have developed. The figure on the right shows some sphere-like, cell-like structures that are called coacervates, and you can form such structures in a laboratory environment, but they're really not living. So the concept here that I want to then expand on is, how did these first living cells, what was their metabolic activity? It is considered that metabolism was very important in the formation of life. You have to have some source of energy to survive. So it's thought that the first metabolic pathways were, number one, anaerobic. They occurred in an environment without oxygen. The best that's been provided so far is that glycolysis is considered to be one of the first, if not the first, metabolic process to develop to form a small amount of ATP, a net of two. It's considered that fermentation might have been an anaerobic process that would occur to allow this electron carrier called NAD to be regenerated such that glycolysis could continue. Both processes occur in the cytoplasm of the cell. That's important just from the standpoint of there's no organelle where these processes occur. They just occur out in the cytoplasm of the cell. Even in our cells, they do. So the assumption here is that a consumer or a heterotroph would have evolved first before a producer that would have some pigment like chlorophyll or some way of producing sugars from from molecules that it would absorb from the environment. So let's look at this process of glycolysis again. I put a different figure in this video. You might notice that glycolysis is a 10-step, linear, enzyme-driven process, starting with glucose most commonly, and ending with two molecules of pyruvate. There is an expenditure of 2 ATP. They are in the pinkish boxes at steps 1 and 3. There is the production of 4 molecules of ATP in the pale green boxes at steps 7 and 10. There is the production of 2 molecules of NADH in kind of a tannish beige box at step 6. For glycolysis to continue, this electron carrier NAD has to be present in the cytoplasm. So without oxygen present, fermentation is a survival tactic in our cells, but in anaerobic organisms, it's a perfectly designed mechanism to maintain glycolysis. So fermentation is very important in the development of life. So you notice that these anaerobic processes have been passed on. It's considered that this passing on of these processes is kind of a support of the evolution of life from simple or non-complex, non-advanced organisms like bacteria and yeast to more complex, advanced organisms like plants and animals, the human, primates. So fermentation is a process to regenerate the molecule, the electron carrier NAD, so that glycolysis can continue. Fermentation does not generate any energy or ATP. There are 2 common fermentation pathways. One is called lactic acid or lactate fermentation. It occurs in bacteria and even human cells. It takes pyruvate and forms lactic acid or lactate. You would find that in yogurt, some cheeses, some fermented foods like sauerkraut. You also find lactic acid in muscle cells. If you were to go over to the creek and work out, use the weights, you might find your muscles sore the next day from the buildup of lactic acid. You might notice that in the formation of lactic acid or lactate from pyruvate, 2 hydrogen atoms are added, and that those 2 hydrogen atoms are added from NADH that regenerates NAD such that it can filter back into the glycolysis pathway and continue the production of 2 ATP as the net. The second type of fermentation is called ethanol fermentation. In this regard, you have the production of carbon dioxide and the eventual formation of ethanol. Corona, the finest beer our Spanish word for today. Corona means crown. So think you can look up the importance of beer in history of the world. So the formation of alcohol, beer, wine, and other spirits, if you will, is accomplished by yeast. And you notice in the process, you have the formation first of acetyl or acetaldehyde, and then the formation of ethanol. You have the formation of carbon dioxide that's released. So you actually have a molecule with less carbon atoms than pyruvate, one. So alcohol fermentation and lactate fermentation, both, as you see, regenerate the electron carrier NAD in the cytoplasm so that glycolysis can continue to generate energy for the organisms that have these pathways. Some people utilize a fermentation diet. There are lots of fermented foods and even beverages, if you're legal enough, haha, to enjoy. So I have yogurt every morning for breakfast. I sometimes drink kefir, which is kind of a liquid-like yogurt. They both have active cultures of bacteria that convert some of the lactose sugars, the milk sugars, if you will, in these products into lactic acid. Sauerkraut. Cabbage has been fermented for thousands of years. It increases its shelf life greatly and allows it to be used over a much longer period of time. Sourdough bread is formed by yeast. You see in picture B a machine that is mixing the dough together. In sourdough bread, the yeast release carbon dioxide, and that causes the flour to rise and form bread. Most people today make bread using baking soda, and baking soda releases carbon dioxide and results in the bread, the dough, if you will, rising. In photograph C, you see the individual yeast cells themselves. You see in the yellowish portions, you see them budding or reproducing in an asexual manner. Yeast are used in the formation of beer and wine. They can be distilled and produce a higher alcohol content spirit, such as vodka or gin. Wine is typically thought of as a fermented beverage from grapes. Beer is usually fermented from some grain, rice, wheat, and even others. This last slide talks about, and your book also talks about it, OpenStax talks about how other organic macromolecules such as lipids and even proteins can enter the respiratory process at different levels. Glycerol can enter in glycolysis toward the middle. Some of the fatty acids can enter the Krebs cycle. Some of the amino acids can enter at different steps within the respiratory process and can be oxidized again to produce ATP and generate heat. So it is possible for different macromolecules to be oxidized to release ATP energy, primarily in electron transport chain, step 3. But you might notice the most efficient macromolecules to go through the process are carbohydrates, glucose, and other monosaccharides. So that ends our video set for chapter 7. I will next post chapter 8 in a couple days. So God bless, take care, and see you soon.